So, welcome to all the BSc first year students. Paper first, diversity of cryptogams. Yesterday we have seen that is the thallus structure of Cara. What are the different parts of thallus of Cara? Single cell structure at nodal region, a single cell structure of internodal cell. So details regarding all these things we have seen yesterday, we have studied yesterday. Now, today we are going to be studying that is how the multiplication takes place or reproduction takes place in Cara. Already I have mentioned that multiplication or reproduction in case of Cara take place by only two ways. There is no asexual method still there is no discovery of asexual method of reproduction. There are two ways by which the Cara multiplies or reproduce, that is vegetative methods and sexual methods. So before going to be study the different structural organs of vegetative method of reproduction and sexual method of reproduction, what is meant by vegetative method of reproduction? Vegetative method of reproduction means without forming any gametes or any part of the plant body or a special structure, vegetative structure which are produced by that particular organism, from which a new organism develops, then it is called as vegetative method of reproduction. Any part of the plant body, for example leaf, stem, root, from which or there are certain outgrowth which comes on older plants, on parental plants. From that outgrowth again there is a formation of a new organism or new plants, so then it is called as vegetative method of reproduction. That is known as vegetative method of reproduction. So what are the different structures from which vegetative method of reproduction take place in Cara? There are three types of structural bodies develops on rhizoids of Cara, that is while vegetative method of reproduction, that is amylum stars, stars, a star-shaped structures which are having a large amount of amylum star, a large amount of food material. The structure is star-shaped, therefore they are known as amylum stars. These structures are formed on a parental plants of the Cara. When they detach from the parental plants, from which again there is a formation of new Cara plants, then it is called as vegetative method of reproduction by producing amylum stars. There are again certain spherical round bodies are formed on a rhizoids of Cara. And the conditions are favorable, such bodies are formed. So they are known as bulbules. They are again, when they detach from the parental plants, from that again there is a formation of new Cara plants. Then another, a small filament structure or outgrowth produces, and that outgrowth is known as protonema. So that protonema is also a vegetative reproductive bodies. They are also again, when they detach from the parental plants, they turns to formation of a new Cara plants. So these are the three ways by which vegetative method of reproduction take place, by producing amylum stars, bulbules and protonema. These are the special major vegetative reproductive bodies of Cara. So this is the vegetative method of reproduction in Cara. Now the most advanced complicated method of multiplication or reproduction in case of Cara is sexual method of reproduction. While describing before going to study the structure and development of sex organs, that is male reproductive body and female reproductive body in Cara. First of all, I have to give a slight introduction regarding what type of, how the sexual reproduction takes place in Cara. So, the sexual method of reproduction is very complex, complicated, with well-developed sex organs, oogamous type of sexual method of reproduction, and the reproductive bodies, which are very specific one and common one. They are macroscopic. Means we can be observe them without microscope. We can be observe them naked eye without microscopes. So they are known as, therefore they are known as macroscopic reproductive bodies in case of Cara. So, and their position is very specific one. So the nucleus is just above the globule, means the position of these reproductive bodies, that is nucleus and globule, is very specific. Then you have to write down a side reading of structure of globule, structure of nucleus. So while studying the structure of globule, it is a spherical, round in nature. The outermost wall is made up of from 8 shield shield cells. So these are the 8 shield cells, curved 8 shield cells. This is the outermost coating. Inner that shield cells, there are a dumbbell shaped specific structures are there, which are originated from the center of each shield cells, that is known as the manubrium. From the center of shield cells originated a dumbbell shaped cells which are projected toward the center of the globule. It is known as manubrium. Now, the tip of these manubriums cuts into small rounded or hexagonal structural cells. They are known as primary protonema. Again, from that primary protonema, small structural cells are originated. They are known as secondary protonema. So, primary, secondary protonema. From that cells, elongated multi-cell structures, filamentous structures are produced. They are known as antheridial filaments. So these antheridial filaments is having a number of cells. Then each cell there is a prominent nucleus. From that, flagellated structures are produced in each cell of the filament. So this is the each cell of antheridial filaments. From that each cell there is a formation of biflagellated pear-shaped structures, that is known as anthrozoids or male gametes, which is motile one. So on the maturity of these globule, it bursts, opens, filaments are exposed. From that antheridial filaments, they opens, and from each cell of each antheridial filaments gives rise to a number of bioflagellated structures, anthrozoids or male gametes. In this way, that is the structure, the formation of anthrozoids or male gametes, details regarding this of globule. So similarly, female reproductive body here is known as nucleus. A female reproductive body is known as nucleus. Nucleus is a elongated pear shape, and it is made up of from a number of 5 to 8 tubular cells, which is having a centrally located prominent nucleus which is act as an egg, and terminally these two cells cuts into a small tongue-shaped structure which is known as corona. So on the maturity of these nucleus, small silt or opening is formed in that corona, through which whatever the motile male gametes are formed, they gather at the point of silts or opening, through which there is entry of number of male gametes take place. But out of these number of male gametes, any one male gametes unites with the egg, and that process is known as fertilization. After fertilization or after union of male and female gametes, so now this nucleus turns into black in color. So on the dead and decaying of these parental plants, so these black structures which are diploid in one, to a number of chromosomes diploid in one, is act as a oosphere or zygote. So that zygote is abundant in the soils, and it remains dormant in adverse condition on the onset of rainy season. So by breaking up the wall of oospores or zygotes, there is a germination, or that diploid nucleus divides meiotically and gives rise to 4 haploid nuclei. Out of these 4, 3 haploid nuclei degenerates, or from that there is a formation of origination of a new charaplants In this way, sexual method of reproduction take place in case of chara. So in examination, remember that in examination there will be a 10 marks questions on the sexual method of reproduction in chara, or sometimes a 10 marks question on structure of globule or structure of nucleus. Or 10 marks questions, or they may be asked a 5 marks questions on structure of globule or structure of nucleus. 10 marks or 5 marks. So again there will be a 20 marks questions on chara, that is, give a detailed accounts on life cycle of chara, or describe thallus structure and reproduction in chara. Both are same. So you have to write down details regarding the life cycle. That is, first of all you have to give the classification, occurrence, structure with figures, and reproduction or multiplication in case of chara. Then you have to write down a graphical representation of life cycle of chara. So with this we have completed the chara and their life cycle, and in the next class tomorrow we are going to be dealing about another. So tomorrow we are going to be dealing the next class, that is Xanthophyceae, or they are also known as yellow algae. In that they have been mentioned for your syllabus to study the life cycle, that is Botredium. So here also you have to be describe the detailed life cycle of that particular example, that is Botredium. Then here also you have to first give the classification, occurrence, structure, and reproduction. So that is the only one examples they have been mentioned for your syllabus to study the as a type study or example from Xanthophyceae. So they are commonly known as yellow algae. Why they are known as yellow algae? So details regarding tomorrow we are going to be dealing about that. Thank you, one and all. Okay, thank you.